what I'd like to do for the next few minutes is just review the status and the prospects for the development of a chikungunya vaccine. And if you think about vaccine development, there are always a number of challenges. And for chikungunya, I think that there are several factors that will facilitate vaccine development. For example, there's only a single chikungunya serotype, no evidence of reinfection. There are well-established correlates of protection for alpha viruses, mainly neutralizing antibodies. And we can manipulate alpha viruses genetically very simply, and I'll give you an example of that in a moment. But there are also some major challenges. First of all, with animal models, wild-type rodents are very poor models for chikungunya. Adult immunocompetent mice basically generate little or no disease and are unuseful for vaccine testing. There's unpredictable incidence of chikungunya, and this is a major challenge for efficacy trials where typically a vaccine is tested in a susceptible population that's exposed to the disease. So some people are vaccinated, some people receive a placebo, and we look for a difference in disease incidence in those populations. But if we don't know where chikungunya will be or how much incidence of disease there will be, it's very difficult to plan for these studies. And finally, another route to showing efficacy that was recently implemented in the US by the Food and Drug Administration called the animal rule, where you can show efficacy in a very reliable and authentic model for human disease just by experimentally testing animals. In theory can be used for chikungunya, but has not yet been used to license any vaccine in the United States. So in my thinking, the ideal chikungunya vaccine would be simple and inexpensive to manufacture. It would induce very rapid yet long-lasting immunity after a single dose, and I think a single dose is very critical for intervening during an epidemic. It would be stably attenuated in the case of live attenuated vaccines with virtually no chance of reversion to virulence. And another important safety consideration if you're going to use a vaccine in a non-endemic location would be that it cannot infect mosquitoes and initiate a transmission cycle, which could be very unpredictable in how the virus would evolve, possibly revert to virulence. So this is the current situation of various vaccines that are underdeveloped. I won't go through all of these for you today, but you can see that there are a lot of them in preclinical phase or in animal testing. There are two that have recently undergone phase 1 clinical evaluations. These are very small studies just to show safety in people not necessarily exposed to disease. And there's one vaccine that underwent phase 2 testing about 30 years ago. So various platforms have been used to develop these vaccines, including live attenuated, inactivated wild-type virus, virus-like particles, DNA vaccines, and as I'll show you in one example with measles, using other viruses as vectors for chikungunya vaccines. And I'm going to talk about 4 examples of these. One of them is shown here on the right that was developed by the US Army in the 1980s. It's called 181 clone 25. This strain proved to be very immunogenic in volunteers who received it, but also to be a little bit reactogenic. Some of these vaccinees had mild arthralgia, and this program was discontinued by the Army about 30 years ago. But this strain is a very useful benchmark for us to use to try to match its immunogenicity, which is excellent, but reduce this reactogenicity and make a little bit safer vaccine. So I'm going to show you some comparisons of our newer approach with this vaccine, which we now know is attenuated by 2 simple point mutations, which is probably the explanation for its reactogenicity, because it can revert very easily to virulence. So the measles vaccine recently completed phase 1 testing in Austria. Basically, the vaccine strain of measles in cDNA form is genetically engineered to include the structural protein genes of chikungunya virus. Then this vaccine can be produced in vira cells. It also, when it replicates, produces wild-type proteins, but is limited in its replication, presumably in people and in animal models, so that a single immunization can protect mice from lethal challenge. And as I'll show you, the phase 1 data indicate that 1 or 2 doses in humans is highly immunogenic. They've also shown that immune sera from mice that receive this vaccine confer protection, again showing that antibodies are the main mechanism of protection with these vaccines. So in the phase 1 study, which was just published this week in The Lancet, 42 volunteers were immunized. It was safe and well tolerated with minimal side effects. With a single dose of the vaccine, over 90% of people seroconverted in the medium and high dose groups. The titers, though, which were measured as 50% neutralization titers, were relatively modest at about 48 to 46. And after a second dose, those titers increased quite a bit. And there was no indication of interference with pre-existing measles immunity, which was one concern about this vaccine. Since most of us have antibodies against measles virus, they would prevent the efficient replication and expression of these chikungunya proteins. The second vaccine was developed at the NIH's intramural laboratory in Bethesda, Maryland. This is a virus-like protein vaccine where a plasmid DNA is engineered to express the 3 main structural proteins of chikungunya virus. And then when cells are transfected with RNAs from these clones, they produce virus-like particles that are essentially identical in structure to chikungunya virus, but they're completely incapable of replication. So they serve more or less like an inactivated vaccine preparation. And these have been tested in mice and eventually in monkeys. They induce high titer neutralizing antibodies that protect monkeys against viremia. And once again, taking sera from those monkeys, these investigators were able to show that those sera, when transferred into mice, protected against disease. So humoral immunity is again the mechanism of protection. So this vaccine also went into phase 2 trials. Again, a small group of people received 3 different doses from 10 to 40 nanograms of DNA, which is quite a large amount of DNA. It was well tolerated again. Neutralizing antibodies were detected in all of the vaccines after 2 doses. So this is a vaccine that appears to require 2 doses for efficacy. Again, the 50% neutralization titers, which is a very, let's say, liberal estimation of immunity, were much higher, though, than with the measles vaccine. After 2 doses, they were well over 1,000. Unlike the measles vaccine, which is licensed to a company in Austria, this NIH vaccine, which was originally licensed to Merck, is no longer under a commercial license. And unfortunately, in order to develop a vaccine to licensure and marketing requires hundreds of millions of dollars typically. So this vaccine is going to have to pick up another commercial partner to move forward. So finally, I want to mention a little bit about our vaccine. This is a live attenuated vaccine made with a chikungunya virus genome. So as Christine showed you the organization of the genome on the top panel here, the virus includes a subgenomic promoter. It makes large amounts of a subgenomic message that encode the capsid and E2 and E1 envelope glycoproteins. And in order to attenuate this virus, what we've done is we've eliminated that subgenomic promoter with 13 synonymous mutations to completely knock out its activity and eliminate the possibility of reversion. And then we substitute it with an internal ribosome entry site from a picornavirus, which allows the structural protein genes to be translated from the genomic RNA itself rather than the subgenomic RNA. But this greatly downregulates the expression because normally when alphaviruses replicate, they make much more subgenomic than genomic RNA. And a second version shown at the bottom, we leave the promoter intact, but we swap the position of the capsid gene downstream of the envelope glycoprotein genes and put the capsid behind the iris. So in this case, only the capsid is expressed via the iris. Now, the other reason we included this iris is that this is an iris that cannot be recognized efficiently by ribosomes in insect cells. And so these 2 viruses, version 1 and version 2, are completely defective for replication in mosquito cells and cannot infect mosquitoes at all. We think this is an important safety consideration. So as expected on the left panel, there's no subgenomic RNA present in iris version 1. There is in iris version 2. As shown on the upper right panel in green, although This vaccine strain replicates more slowly than wild-type chikungunya, and compared to the 181 vaccine strain as well, it ultimately reaches titers a little over 10 to the 7 plaque-forming units per mil, which is sufficient for efficient production at a large scale for vaccine production. Now, we've looked very closely at the safety of this vaccine, and again, we've compared it to this 181.25 strain developed in the 80s. And these are some studies we did with A129 mice. These are mice that are defective for their interferon type 1 receptors, so they develop lethal disease with wild-type chikungunya virus infection, and they're very useful for studying the efficacy in an inexpensive small model like this. And what we've done is we've compared our iris vaccine on the upper right to the 181.25 upper left, and then wild-type chikungunya. We've looked at a large number of different organs shown across the bottom of these graphs, and then we've looked at a time frame from 1 to 8 days after infection. And what you see on the bottom is that in these mice, wild-type chikungunya very rapidly produces a systemic infection. There are high titers of virus in virtually every organ, including the brain in these animals. The 181.25 strain also produces a systemic infection a few days after inoculation, but it's slowed down compared to wild-type, and the titers are reduced a little bit. By comparison, our chikungunya iris strain on the upper right produces virtually no replication in any tissues other than the muscle, spleen of the animals, and again, very low titers. An even more severe test of the safety of this vaccine is to look at the stability of attenuation. So what we've done here, again comparing to 181.25, we took these vaccine strains and passaged them serially intracranially in baby 181.25 mice, the most permissive environment we know of in vivo for these strains. And then we looked after 5 passages to see if their virulence had increased. And what we found, first of all, if you look at wild-type chikungunya on the left here, that strain kills all these mice by day 4, baby mice. When we look at the 181.25 strain, it began this study completely attenuated, did not kill any mice, but in 2 independent passage series, by passage 5, you can see that this strain becomes completely fatal, killing all of the mice by day 13 in 2 different independent experiments, whereas the iris strain remains completely attenuated, doesn't kill any mice after 5 serial brain passages. So this is a very high degree of stability for a live attenuated vaccine. We've also looked at the correlates of protection, and once again, as I mentioned for the other vaccines, when we take sera from vaccinated mice and we transfer them passively into A129 mice and then challenge those mice with wild-type virus, at the undiluted or 1 to 5 dilutions of the serum, we get complete protection from the antibodies. And then as we dilute the serum further out, we get less and less protection, eventually reaching the same as normal mouse serum, where there's no protection at all. So again, it's the antibodies in the serum that are protecting the animals. Finally, we've done a large number of studies with cynomolgus macaques at the Tulane Primate Center. The basic design of our experiments is shown here. We implant these animals with telemetric devices that almost continuously monitor their heart rate, their respiratory rate, and their core temperature. Then after they recover from the surgery, we vaccinate them. Our vaccinations are all a standard dose of 5 logs of virus. We bleed them to see if the vaccine produces viremia, which it never does. We bleed again at several time points to measure their antibody response. And then anywhere from 45 days to a year after a single dose, we challenge them with wild-type virus. This is not a lethal model. Like in people, chikungunya is not normally lethal. But these telemetrically monitored signs of disease are very useful in showing protection. And then we also bleed to look at viremia after challenge. So what we find in these experiments is that the animals by day 45 develop very robust neutralizing antibody titers. We use the more conservative 80% cutoff for our strains. We did 3 different experiments using either subcutaneous or intradermal vaccination with either the version 1 or version 2 vaccine strains. It seems like subcutaneous maybe is a little bit more immunogenic. And then when we monitor these animals after challenge, what you're looking at here in red are sham immunized animals, which immediately after challenge develop fever for about a day and a half, shown in the red peaks. But then, as you can see later on, they become severely hypothermic. And this is probably due to their inactivity because they're suffering from severe arthralgia. But the vaccinated animals in the two different groups shown in green and blue are completely protected from fever and from hypothermia, and they maintain the normal diel periodicity of their temperature profiles. More recently, we've tested this vaccine against a Caribbean strain of the virus. The reason is that the vaccine is made against the La Réunion strain from the Indian Ocean lineage, and we wanted to make sure to protect against the Asian strains circulating here now. We did experiments in A129 mice where we showed that against at 3 different doses, our vaccine protects both against weight loss and against swelling of the inoculated foot pad. And then recently we completed studies in cynomolgus macaques where we again immunized and challenged with a Caribbean strain from Guadeloupe in January of last year. And we showed that in the case of the Caribbean strain, we see a little bit less of the hypothermia in the sham vaccinated challenged animals. You'll notice on the upper graph, those red bars do not extend downward as far in the later stages after challenge. We do see a febrile response that's a little bit lower than the La Réunion strain, but similar in length. And once again, all of our animals were completely protected from fever in these studies. So to summarize the prospects for development of a vaccine, scientifically, I think that chikungunya is a relatively straightforward target because of its limited antigenic diversity and lack of immune enhancement that we always have to worry about with dengue vaccines. The main problems are the financial uncertainty related to the cost of getting a vaccine licensed, where would efficacy trials be done, and what would the market look like in a few years? Is there going to be a profit to be made, for example, here in the Caribbean 5 years from now if a vaccine is licensed? So I think there are several very promising candidates that are poised for either phase 1 or phase 2 trials. And probably in the next few years, if there's a clear regulatory pathway and adequate funding, commercial funding in this case, I think there's a good chance we'll see a chikungunya vaccine available. So I just want to close by mentioning our collaborators at Takeda Pharmaceuticals, who have licensed the technology and are developing this vaccine. At the Tulane Primate Center, Chad Roy leads the team that does the monkey experiments. The people in my lab involved in the testing and development of the vaccine are shown in the upper left. And finally, let me give a great thank you to the Caribbean Public Health Lab for the invitation and the hospitality to come, and my friend and collaborator, Christine Carrington, who I just embarrassed a little bit here, perhaps with this photo, in my last visit to Trinidad when she decided to take a gringo into the middle of the celebrations here in Port of Spain. So thank you very much for your attention.